In this section, we're going to briefly discuss the targeting of other types of proteins, other than those that we've already covered. So we're going to briefly discuss the targeting of transport proteins, structural proteins, and look at targeting protein-protein interaction sites. So first of all, an example of a transport protein. Now, transport proteins carry polar molecules across the cell membrane. So here we have a transport protein with a polar molecule approaching it. The polar molecule enters into the transport protein, which then pushes the molecule to the inside of the cell. So this is undertaken through the process of active transport. Now, the binding site recognizes this guest molecule, the transport across the membrane. And so there was a push to develop drug molecules that could mimic this guest molecule, but instead of being transported across the membrane, they would bind very strongly to the transport protein and block activity. Now, this type of approach can be used to increase the levels of neurotransmitters at nerve synapses. And one example of a drug that acts in this way is Prozac, or fluoxetine. Now, this blocks the transport protein that's associated with serotonin uptake, hence why it's in the class of selective serotonin uptake or reuptake inhibitors. It blocks the transport protein that is associated with the serotonin uptake and stops it from being transported across the cellular membrane. We can also design drugs to target the proteins that make up the structural elements in cells and tissues. Now, this can also include targeting things like viruses and the viral protein coat. Now, this protein coat is a capsid, it's a capsule that coats the outside of the virus, and we can produce drug molecules that are able to target this protein coat. Now, the cold virus is a type of rhinovirus. It's made up of a capsid which possesses 4 different protein subunits, and you can see what the outside of a viral capsid looks like here. Now, a canyon is present within this protein coat that contains or allows access to a hydrophobic pocket, and this can be seen here. Now, what happens is a receptor on the host cell will bind into this canyon, so it will bind into this pocket, and the virus will then undergo or induce a conformational change. And the conformational change will then release the virus. And this virus will be released into the cells. Now, the hydrophobic pocket is occupied by something called a pocket factor. Now, it's thought that this is needed to stabilize the capsid before it's able to bind to the host cell, and that it prevents the conformational change needed to release the virus. Now, this is just an example of the hydrophobic canyon that is present in the virus here. So this region contains lots of hydrophobic residues, so hydrophobic amino acids within it. Now, some antiviral drugs are thought to mimic this pocket factor by binding in the hydrophobic pocket themselves. Now, here is one example of a molecule here that is able to do this. It's able to bind into the hydrophobic pocket that lies just underneath the canyon floor. Now, these are typically very long chain hydrophobic molecules, and these act to stabilize the capsid into a conformation that prevents the release of the viral nucleic acid into the cell. So this drug molecule acts to stabilize this pocket and prevent the release of the nucleic acid. Now, this can also change the shape of the canyon floor, which prevents the virus from binding to the receptor. As it can't bind to the receptor, it means that it cannot then infect the host cell, and therefore it inhibits the viral replication process. Now, viruses are very, very complex molecules to target. This is an example of the viral capsid that is found in the foot and mouth virus. Now, the elements in red are your alpha helices and in yellow your beta sheets. Now, targeting any particular part of this virus, or at least targeting the viral capsid itself, is very, very challenging. As you can see, the capsid looks nearly the same throughout, and it is very, very large in comparison to a single protein. And so this is one of the reasons why developing antiviral drugs is incredibly challenging. Now, another example of a type of protein that we can target is tubulin. Now, tubulin is the building block for microtubules, and these have a variety of different roles within the cell. Now, here we can see tubulin, and this is able to polymerize into these microtubules. So these microtubules are able to offer structural support within the cell and support different cellular processes. Now, during cell division, the microtubules depolymerase to give tubulin, and this is then used to form a spindle. Now, the spindle is used to push the two cells apart and help transfer the chromosomes into the daughter cell. So here we have our parent cell with our nucleus, and during cell division, a spindle is formed that helps to transfer the chromosomes into the daughter cells. And so the tubulin plays an incredibly important structural role in this. Now, inhibiting cell division can be a very useful approach in the treatment of cancer. And so we can produce drug molecules that can either bind to the tubulin to prevent it from polymerizing, or we can produce drugs which stabilize the microtubule itself and prevent this depolymerization process. And again, I just want to show you what the structure of this protein, of this polymerized protein, actually looks like. So this is the tubulin structure. Now again, in red you have your alpha helices and in yellow your beta sheets. Now hopefully you can see that the tubulin contains a very large number of alpha helices, and this suggests that they are very strong and help it to perform its role as a structural protein. Now you can also hopefully see this hole that runs down the middle of the protein. Now, of course, in the cell, this wouldn't be a vacuum. This would be filled with cytoplasm or with water and other cellular components. But hopefully you can see if you were to extend this structure, you would get something that looks like a rod or perhaps like a rope. And it's its structure that is key to the function that it performs within the cell. Now, we can also try to design drug molecules to inhibit the interactions between multiple proteins. And targeting these protein-protein interactions may allow us to inhibit the biological processes that these proteins take part in. Now, the regions of interactions between different proteins can be quite large, and these can often contain 40, 50, 60 or even more amino acid residues. And it's these amino acids, or rather their side chains, that interact with each other. And so these interactions can help to hold proteins together. They can be formed between multiple protein subunits to make up a functional protein molecule. Now, to target these regions would require very large drug molecules because the regions themselves are large, and these molecules would have great difficulty in crossing the cellular membrane. However, research has shown that often it's a few key amino acids that are important for a large percentage of the binding interaction. So we could try and design drugs that target these key amino acids as a possible new way of targeting these interactions. Now, one example of an important protein-protein interaction is between the two proteins p53 and MDM2. Now, p53 is involved in cell growth and death in cells which are under stress. And therefore p53 acts to help protect the body from defective cells such as those that have become cancerous or faulty in other ways. Now, in some tumour cells, the protein MDM2 is overexpressed and it acts with p53 to inhibit its protective function. Now, molecules which interfere with this action could be useful as potential anti-cancer drug candidates. Now, the compound nutlin-2, which you can see here, interacts or mimics three important amino acids on the p53 protein. So it mimics these three amino acids here. Now, nutlin-2 is able to bind to the region of MDM2 that is normally involved in the interaction between MDM2 and P53. And so if we introduce nothing 2, we can use this to block the region of MDM2 that normally interacts with P53 and allow P53 to perform its normal function. However, this is a very complex process, and drugs which block protein-protein interactions are quite unusual and quite rare. Now this brings us to the end of the Structure and Function lecture series. Now in future we will have a workshop where you'll be able to have a look at questions related to content within the Structure and Function lecture course, and you'll have a chance to have a go at these questions and review where you are particularly strong and where you need to improve. Now on this slide is a list of the main areas that were covered within the Structure and Function lecture series, and you will see these reflected within the questions in the workshop. Now if you have any questions about this particular content, please don't hesitate to post on my support forum.